Hello students, in previous part we have discussed about eukaryotic and prokaryotic cell. In this section we will start some cell organelles, and the first cell organelle is cell membrane. Cell membrane. Cell membrane is also called biomembrane as well as the plasma membrane. Plasma membrane, cell membrane or biomembrane. Firstly, what is the component which makes the cell membrane and what are the properties of the cell membrane? Plasma membrane is made up of lipid and phospholipid. You can say that phospholipid and protein. Plasma membrane act as semi-permeable membrane as well as selectively permeable membrane. What is the difference between the semi-permeable and selectively permeable membrane? It acts as semi-permeable membrane. What is the difference between semi-permeable and selectively permeable? Membrane. We know that plasma membrane, this is the plasma membrane, and plasma membrane contains small pores through the substances may in or out. Why the plasma membrane called semi-permeable membrane? This is the inner side and this one is the outer. If there are some substances may present in the atmosphere, these likes, this plasma membrane allows some substances to pass through in and out. So it is called semi-permeable. For example, if these particles when enters into the plasma membrane, the small particle may pass out through these pores, but not these substances. But there are no sure all these small particles which passed out through the plasma membrane are useful for plasma membrane or not. Some harmful substances may also pass through these pores. So the particles which pass through the plasma membrane through the pores, those are small in size. There is no separately which are harmful and which are useful. All these may pass out. So it is called semi-permeable membrane. But in selectively permeable membrane, why the plasma membrane called selectively permeable membrane? Same, this is the plasma membrane, it contains pores and the particles is here, these like. The plasma membrane only allows those substances which are useful for the plasma, which are useful for the membrane. Only allows those substances. Maybe this substance is smaller. This substance is smaller than the pores, but the plasma membrane doesn't allow this substance to enter through these pores. It allows only those chemical substances which are useful. Maybe if this substance is required by the plasma membrane, then plasma membrane makes a fold and try to engulf or try to take these substances through this fold, and it is utilized by the plasma membrane. It allows only the required material to pass through in, so it is called selectively permeable membrane. But through the pores all the substances may be useful or harmful enters, then it is called semi-permeable membrane. What are the properties? Now the properties of plasma membrane. Properties of plasma membrane. The first property, plasma membrane allow the fatty and water molecules through this layer. It allows the fatty particles as well as water molecules through it. This is the first property. If plasma membrane allows these substances to pass in, that means it follow the properties of plasma membrane. Second one, the phospholipid and the protein, because we know that plasma membrane is made up of phospholipid and protein. The ratio of phospholipid and protein may be variable. Ratio of phospholipid molecule and proteins variable. It means changeable, because if these particles may be changed, then the plasma membrane remains in kinetic mode. Number third, it remains in kinetic mode. Kinetic mode. These are the properties. If plasma membrane, if any membrane allows these properties, that will also follow the rule or properties of plasma membrane. On this basis, so what is the structure and model of plasma membrane? The plasma membrane model was proposed by different scientists. And what were the different postulates which were proposed by the different scientists? Those are, the first plasma membrane model was proposed by Danieli and Davison. Model of plasma membrane. Plasma membrane. The first plasma membrane model was proposed by Danieli and Davison. They proposed the plasma membrane model. They compared the plasma membrane model with the sandwich model. Sandwich model. What is sandwich? We know that there is two slices of bread and which we can keep the jams just like that. First, the plasma membrane proposed by Danieli and Davison, and the model which was proposed by those two scientists called sandwich model. What were the properties of sandwich model which was proposed by Danieli and Davison? Firstly, plasma membrane is made up of protein and phospholipid. If it is made up of protein and phospholipid, that is a plasma membrane. This was the first one. Second, proteins are present in alpha globular forms, means proteins present in globular forms in the alpha globulars. Phospholipid is a part which is made up of diglyceride. What is diglyceride? It means a head. It contains glycerol head and two fatty chains are attached with the glycerol head. That is called phospholipid diglyceride. Phospholipid is amphipathic. What is amphipathic? Phospholipid, it means one glycerol head. This is a glycerol head, and it is attached with the two fatty acid chain. These are the fatty acid chain, and one side it is attached with the phosphate. One side it is attached with phosphate, so it is called phospholipid. This is the structure of phosphodilipid because bilayer of lipids. Third one is plasma membrane. Plasma membrane is made of two phospholipid layer, or we can say that biphospholipid layers. Amphipathic. What is amphipathic? Amphipathic means one end which doesn't react with the water and another one that reacts with water. It means one end dissolved in water and other one not. Glycerol head is the hydrophilic. Hydrophilic, it means it reacts with water. And tail is hydrophobic. Hydrophobic means it doesn't react with water. So one particles which show different mode. One end hydrophilic, another one is hydrophobic. So it is called amphipathic. Clear? And number 5, the proteins are present. The proteins are present outer side of phospholipid. And 6th property which was proposed by Daniel and Devson, the bilipid layer. The tail face and both ends each other, but the head remains away from. In phospholipid, the tail face each other and the head away from each other. It means tails are one to one, head to head present, but the head is away from each other. And last one is the tails are attached with each other with a weak force, and that force is called van der Waals force. The tail are attached with a weak force. That force is called van der Waals force. These were the property which was proposed by Danieli and Devson. According to this property, we draw the diagram of the plasma membrane. As the Daniel and Devson proposed that plasma membrane is a bilipid layer. Bilipid layer means such like this one. One is the another one side, and another one head is opposite to other and tails face each other. This one like. Tails are face each other and head are opposite in opposite direction. First one, this like structure. This is the head and these are the tails, and they makes the phospholipid bilayer. Next, the protein present in globular forms and outside the phospholipid. This is the protein. Why the Daniel Devson proposed that the sandwich model? This protein makes a two layers just like a bread slice, and these the substances gems like present inside it. So they called it sandwich model. These are the proteins. They showed this one like structure, means protein present in globular forms, alpha globulars, which are present outside the phospholipid, and phospholipid bilayered in which tail face and each other and head opposite to each other. tails are attached with a weak force that is called van der Waals force. Head act as hydrophilic, reactive with water, and tail hydrophobic, that doesn't react with water. So the phospholipid act as amphipathic. Clear? This model was proposed by Danieli and Davidson. Now let us, if this model show the properties of plasma membrane or not. Firstly, we'll take the fat molecules or fat. If the fat molecules may pass in through it or not. Fat molecules comes from here. Protein doesn't create any problem to pass any substances through it. So it smoothly pass through it. Then head is the hydrophilic, there is no reaction can take place, and tail is fatty acid, that is a fatty acid, and fat molecules comes here, so there is no problem and it pass through the plasma membrane. The fat molecules may pass through the plasma membrane. It follows the properties of plasma membrane. Second one is the water molecules. Water molecules may pass in or not. The water molecules pass through the protein. Doesn't create any problems to pass through any substances, so it pass through it. Then head is hydrophilic, means it reacts with water. There is no any problem may create by the head, but this area which contain fatty acid, this doesn't allow the water molecules to pass through it. So water molecule cannot pass through it. So we can say that the properties of plasma membrane doesn't matter. This is the first one. Water molecule doesn't pass through it. So it does not follow the properties of plasma membrane. First one. Secondly, the protein and phospholipid show variable ratio. Variable ratio means the particles of protein and phospholipid may be changed. They change their position. We'll take the example of this one. This part. And this part contains how many ratio of phospholipid and protein? Phospholipid 1, 2, 3, 4. There are four phospholipid. Protein 1, 2, 3, 4, 5, 6, 7, 8. And ratio of protein is 8. But there is no any movement can be shown. If there is no movement can be take place, then we can say that the ratio of protein and phospholipid not variable. We know that the property of plasma membrane, the ratio of protein and phospholipid may be variable, but in this there is no change in ratio. And third one, if the movement of proteins and phospholipid doesn't take place, then we can say that plasma membrane is rigid. But we show that plasma membrane show kinetic movement. Here is plasma membrane is rigid structure. So the model of Danieli and Davidson doesn't match with the property of plasma membrane. It was not accepted. After this model, another scientist, Robertson proposed some another features of plasma membrane. What were the features which were proposed by Robertson? And Robertson called the model unit membrane model. Unit membrane model, which was proposed by Robertson. There are some changes in the Robertson model of the plasma membrane. First one, he called it plasma membrane is made up of protein and phospholipid. There is no change. Second one, proteins are present according to Daniel Davidson, alpha globulars, but according to Robertson, proteins are present in beta fibrous structure. Beta fibrous structure. Third property remains same. Phospholipid is amphipathic. It means one end is hydrophilic and second one is hydrophobic. And number 4, plasma membrane is made up of bilayer phospholipid layer. There is no change. The proteins are present outer side of plasma membrane, your phospholipid. That is also similar according to Robertson and Daniel and Davidson. And next one is in phospholipid, the tail face each other and the head away from each other. That is also follow. Next one, tail are attached with a weak force, van der Waals force. There is no more change in the Robertson model as well as Daniel and Davidson, but one is changed. Proteins are present in beta fibrous structures, but in according to Daniel and Davidson, proteins present in alpha globular structure. Now we draw the structure of Robertson model. Robertson model, according to it, there are phospholipid bilayer membrane, the phospholipid bilayer membrane, and protein present in beta fibrous structure outside. Beta fibrous structure. This one like. This model was proposed by Robertson. Then what is the difference between the Danieli-Davidson and Robertson model? Only single protein in Danieli and Davidson present were alpha globular forms, but in Robertson beta fibrous structures. This one. Now can we see? Now we see the plasma, it allows the permission, or it makes the property of plasma membrane or not. Take the fatty molecules. Fat molecules. The fat molecules may pass through it. Protein create no any problems. Pass through the head, and at last fatty acid means the tail of fatty acid, because it is also made up of fats. There is no problem may be pass. There is no problem may be create to pass out the fat molecules. So fat molecules may pass easily. Then the water molecules, H2O. H2O pass from here, the protein creating no any problems to pass any substance. Through head it is also passed, because head is also hydrophilic, but this region is also hydrophobic, as we show that fatty acid contained and doesn't react with water. So this end doesn't allow the water molecule to pass through it. It also same, water does not pass out. Passed out. Third one, there is no movement can be seen in the phospholipid and protein layer. Phospholipid and protein, if there is no movement, then we can say that. Suppose we can take the example of this one. Phospholipid ratio of phospholipid and protein. Phospholipid 1, 2, 3, 4, 5, 6. And protein fibrous 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 1, 21, 22, 23, 24. The ratio of phospholipid is 6 and protein is 24. But there is no movement between the fibrous phospholipid and protein fibrous. So variable resistance, no variable ratio. And if there is no variable ratio, the plasma membrane doesn't show any kinetic movement, so it is rigid. These three properties doesn't match with the plasma membrane, so this model was also not accepted by the other scientists. At last, in 1972, two other scientists, Singer and Nicholson, they proposed the model of plasma membrane, and that model was called fluid mosaic model. Fluid mosaic model, and this model was proposed by Singer and Nicholson. In 1972. What were the features of Singer and Nicholson's fluid mosaic model? They also show that plasma membrane is made up of protein and phospholipid. This was the according to Daniel Davidson. They showed that this one. Proteins are present in alpha globular forms, just as Danieli and Davidson proposed that. Protein present in alpha globular structure. Phospholipid is amphipathic. There is no change according to Daniel Davidson. And plasma membrane is made up of bi-layer of phospholipid layer. This is also the similar properties as Daniel and Davidson. All these properties are also similar. How this model can be shown? They again showed that the protein present in the plasma membrane is of two types. Daniel Davidson showed that protein present outside the membrane. This is not clearly protein present outside membrane as well as inner the membrane. Present outer or inner the plasma membrane. So protein is of two types. According to Singer and Nicholson, protein in plasma membrane of two types: extrinsic protein and intrinsic protein. Extrinsic protein and intrinsic protein. Extrinsic protein, the protein which is present outside the outer layer of phospholipid layer, outside or outer side at the head of glycerol head, that is extrinsic protein. Intrinsic protein, it is present, it is partially embedded, it means it is embedded into the phospholipid partially or fully. The protein which is partially or completely embedded into the phospholipid. There is another property of the outer protein. Extrinsic protein is attached with the phospholipid with a weak, or we can say loosely attached. Loosely attached with phospholipid. It means extrinsic protein, the protein which is present outside, and this outside protein attached with the phospholipid with loosely. This is the major property. This was changed, which was Singer and Nicholson. And second one is phospholipid show two types of movement. Movement movement by phospholipid. Two types. One is transition movement and other one is flip-flop movement. Transition movement and other one is flip-flop movement. What is a transitional movement? The movement by the phospholipid in the same layer. The movement by phospholipid in same layer, we'll discuss later, and movement by phospholipid in the opposite layer. in opposite layer. So we draw the diagram according to the Singer and Nicholson proposed. First one is phospholipid is bilayer membrane in plasma membrane. Phospholipid, this one like glycerol head with a fatty acid tail, this one like structure. Protein may present outside. If it's outside present, that is loosely packed. This is the protein layer. Extrinsic protein and in globular forms, because Singer and Nicholson show that protein present in alpha globulars. Extrinsic and intrinsic protein may be partially embedded into the phospholipid or completely. Suppose this is the protein which is partially embedded into the phospholipid, and this protein is completely embedded into the phospholipid. One more draw with the diagram of phospholipid layer, this one, and it also contains there are protein, extrinsic protein. Clear? This one is extrinsic protein. And these are phospholipid bilayer. This is also extrinsic. This one is intrinsic protein, and this protein is completely embedded, and this one is partially intrinsic protein. This model was proposed by Singer and Nicholson. Now we will see if this model is matched with the properties of plasma membrane or not. Firstly, we take the fatty acid or fat molecules. Fat molecules may pass through the protein. Protein doesn't create any problems, so it passes smoothly. There is no head problem created by the head, and at last through the tail it passes smoothly from outside to inside. There is no problem may be created. Second, we'll take the example of water molecules. Water molecules may pass through this protein. There is no problem. Through head it is a pass smoothly, but from here it doesn't pass. So water molecule take the pathway from this protein, and we know that protein doesn't pass create any problem. So water molecules may pass through the completely embedded proteins and pass through outside to inside. Third one, we have seen that movement by phospholipid. Phospholipid show two movement, transition movement and flip-flop movement. Transition movement, suppose this one is 1, 2, 3, 4, 5 are the phospholipid layer, and this one is A, B, C, D, E and F. What is transition movement? Transition movements the movement of phospholipid from here to here. It means same layer. This is called transition movement. And a flip-flop movement means the movement of phospholipid from here to this one and different layer. This is called a flip-flop movement. As the Singer-Nicholson showed that the proteins are loosely packed with the head of phospholipid. So protein, if any substance may take from here to another, the protein may take from here. Protein passed out. So movement can be seen by the proteins. Clear. Second one, if the movement, transition movement, suppose कीजिए 1 move from here and 5 from here. One move start to move from here and 5 from here. At last they comes here, and 5 is considered the first position, and at 5th position 1 come here. This is transition moment. And third, same, second phospholipid move, phospholipid, and it start to here. C comes here, 2 comes here. It means 2 from here and C from here, it start to movement this way. This is called a flip-flop movement. First property was matched with the plasma membrane. It passed fat acid, fat molecule and water molecule. This was the first property. Water and fat passed. This is the match with the plasma membrane property. Second one, variable ratio of phospholipid and protein. Firstly, we take the example of this one. Suppose this is the examples here. How many phospholipid? 1, 2, 3, 4. 4 phospholipid. Proteins 1, 2, 3, 4, 5, 6, 7. 7 proteins. As we have seen that if protein takes any substance, it moves from one place to another place. So ratio of protein may be changed. Second one, if one start to, we'll take again 1, 2, 3, 4 and 5, A, B, C. If one start to move from here and 4 start to move here, as they start, the 4 doesn't reach at the first position and 1 doesn't reach at the 4th position. So as we count, as reached here, the ratio of phospholipid changed because 4 doesn't reach here and 1 is also not reached here. So they remains here. Then the ratio of phospholipid changed. Ratio of phospholipid changed and show that they show the variable ratio. That also the match property of plasma membrane. Protein and phospholipid change their ratio. That's the matching the properties of plasma membrane. Third, if the protein and phospholipid change their position, why the movement? So the plasma membrane show the kinetic movement. It contains kinetic movement. That is also third position, third properties of plasma membrane, which is also matched. So we can say that plasma membrane is a kinetic movement as well as it changes variable ratio. It changes the ratio of protein and phospholipid, and water and fat molecules also passed it. So the model of Singer and Nicholson was accepted by all the scientists, and this model is also called proteins in iceberg of protein floats at the iceberg of in sea of lipid. Singer and Nicholson proposed their model is just like as protein floats. Proteins are embedded just like as in sea. There are many huge particles of ice float at the surface of sea water, such like as protein float at the iceberg in the sea of lipid. Lipid के sea वर्ग के अंदर proteins particles जो हैं they are float just like as iceberg. Proteins with the phospholipid, it makes a dense liquid-like substance, dense jelly-like substance. So it is called quasi-fluid. Quasi-fluid. In fluid mosaic model, the protein and phospholipid, it makes a dense fluidic-like structures, jelly-like substance that is called quasi-fluid. This model was proposed by Singer and Nicholson, and it was accepted by all the scientists all over the world. In next part, we will discuss our another cell organelles.